Everyone can see you, I just made it so that.
You can't see us.
Oh, okay, great, okay, great, all right, so, thank you everyone for coming for Taniel Winter's Defense. I have a few notes about her. She did her bachelor's in bioengineering and master's in robotics at the University of Pennsylvania. We get lots of great students who come through that route. And started her PhD, Georgia Tech at Emory, biomedical engineering, where I met her while I was on sabbatical. I wasn't even in town, so I was getting like messages from students, and I don't know, I can't. And Taniel went around and looking for various labs, and so like, this is just a good story for everyone to know. She didn't find exactly what she wanted, and so, we talked after what would typically be the next date about what she wanted. I flew back to Atlanta for UNS, and we met like on a table in the conference center or something like that. And it was and I think she was a little bit deflated and like, oh yeah, sorry. Usually the room mics are quite good. Oh, you know what, okay, so anyway, so I think she was deflated. I was like, I don't know if I'm taking a student or something, and we decided to try and to go ahead and make it work, and I was like, okay, but I'm going back to Chicago, and I won't be back till January. So that's how we started, and we had a project from NSF for human-robot interaction where we were doing gait dynamics modeling using switching linear systems, which Taniel worked on and then decided, this is not gonna work, Lena. Like, okay. And I was worried if this project was really aligned with her interests, and little did I know then that she had already written an NSF proposal on locomotion and spinal cord injury without my knowledge, and then she was awarded that NSF scholarship. I mean, she had done it before, I think before we met her. So it just was really well aligned, but I didn't know it, maybe she knew it. And so she wanted to do machine learning. I was like, that's great, I don't know that much machine learning. So of course, Taniel went and started what we now call the Tri-Lab collaboration, and recruited Gordon Berman from Emory Biology and Tricia Kazar in rehabilitation medicine and physical therapy. And she basically just drove the program because we're like, I don't know what to do. She's like, okay, this is what we're gonna do. So she went around to 3 different labs on her own, scheduled everything, very self-directed, very self-motivated, and basically came up with the framework that you will hear today. And this is very unusual for a student to really take that. I mean, I have lots of great students who started projects, but this one was really like, usually I plant a seed, and she was like, that seed is bad. So, and she very soon became, we had a pandemic, and during that pandemic, she was selected for like oral presentations at like very diverse conferences. Physics conference, dynamic walking and biomechanics conference, neural control of movement conference. Then afterwards, she also got oral talks at a posture and gait conference, a neurorehabilitation conference, like every kind of conference. So at Dynamic Walking, we virtually met Michael Rosenberg, who then joined our team, and we started growing like a real project. And both Taniel led with getting her work funded by the NIH as an F31 after her NSF, which then provided seed materials for Michael, the postdoc, to write his F32 that was funded. And they've really been, together carrying this really great project. So this is the first student who was like, please, if I go to a conference, can I give a poster? really want to give a poster. I've never done that. And so she did it at the Disability and Rehabilitation Engineering workshop at USC, where it was received, the work was received really well. And this is just a tour de force project. She got her paper out and people use the code right away. But, it's not easy. There was lots of code, lots of rewriting code, lots of switching code from MATLAB to Python, lots of we're not using that toolbox, let's write our own. And we're done. Oh no, let's rewrite all the code. Right, that happened. Okay.
Exactly.
So you're gonna see this and it's gonna look really smooth and nice. But one thing about Taniel is she's always making things better. You think something's really good and then she makes it better and then you're like, that's great, and then she makes it better. And so she always takes critiques with just to heart and doesn't focus on the negative of it, although, and her paper, when you write a big paper on a new technique, you have to validate everything. You don't have another paper that you have to replicate. So when you guys are asked to just replicate another paper, that's the easy part.
Not to mention that you do it 3 PIs.
Yes, so and then 3 PIs, all who want different parts replicated or validated, and who all think different things are important. And but beyond that, Taniel's just great in the lab, always willing to help, really organized, and does a great job mentoring and setting up understandable tasks for high school students, undergrads, grad students, anyone who's sort of on the team. And we were really lucky last year that as part of our lab cooking gathering, she cooked us, she doesn't cook, she prepared, and then we helped cook. So a whole Trinidadian menu of food for 20 people, which, and again, she took this in stride. The first time we were supposed to do it got canceled because of COVID. In my house. So that's Taniel just persevering and we have to do it later, great. We have to do it again, awesome. I have to cook, acquire all those vegetables, sure, I'll do that. It's a parallel to her academic journey, which has been highly successful. She has fellowships and recognition, and also went on an internship to industry based on one of her presentations, where she also worked on machine learning and Gait. So, and you built a whole program, and we're excited to hear you talk about it. And we will miss you. So.
Oh, I miss you too.
Good morning, everyone.
Good morning.
Thank you, Lena, for such a wonderful introduction. I almost cried, trying to hold it up. But thank you, everyone who is online and who came today in person to take part and listen to my PhD thesis oral defense today. The title of my thesis is Gait Signatures, Data-Driven Discovery of Individual-Specific Neuromechanical Dynamics. So gait is the manner or pattern by which we walk. And you could think of it like almost your fingerprint, where it's unique to you and it could tell your very own story. However, current gait metrics fail to quantify the unique aspects of individuals' gait. And gait analysis is crucial for the identification of gait abnormalities, gait monitoring, and the development of gait rehabilitation strategies towards improving health. The overarching aim of my thesis is to leverage machine learning to help parse complex gait data towards developing tools that can identify individual-specific gait signatures for potential clinical use. And in the end of this presentation, I touch on some proposed future implementations of gait analysis in healthcare using this framework. So gait is important, and it's important because it's an aspect of mobility that impacts quality of life, your safety, and the ability to perform everyday activities. Changes in gait may have clinical implications, which may affect individuals' ability to perform these daily activities. And changes in gait can also be indicative of risks for certain health conditions, such as knee osteoarthritis, or pain, injury, and even changes in disease progression like Parkinson's disease. And if I haven't yet convinced you of how important gait really is, researchers have considered gait speed to be the 6th vital sign. So it's right up there with pulse rate and respiratory rate. And I will focus on the stroke cohort as an example here. So here I'm going to show you 2 stroke survivors. So they've both experienced a stroke, which is damage to their brain, subsequently affecting their motor control. However, their gait disorders are quite unique. So in this left video, I hope to show you, so now I want to play. Okay, let me see. There we go, thank you, Lena. So this video on the left, this individual's impaired leg, which is closer to us, is displaying circumduction. So that leg is moving outwards to clear the floor as he progresses forward. Now the video on the right, this individual does not play that characteristic circumduction. However, the impaired leg, which is further away from the screen, actually has limited heel contact at early stance phase and poor push-off at late stance phase. So we as humans often take walking or gait for granted, as we could typically walk and talk, walk and eat, and even walk and text. I know some of you know yourselves here. I am definitely guilty of that. But the execution of such motion is by no means trivial. Gait requires coordinated movements of various body parts to propel the body forward while still maintaining balance and stability. It first involves the decision to move, which originates in the motor cortex, and neural signals that then pass through the central nervous system, activating precisely motor neurons and coordinating the muscles. Furthermore, the nervous system continuously monitors and integrates sensory feedback from proprioceptors, which are found in the muscles, tendons, and joints, to maintain balance and adjust movement in response to changes in walking condition and even environmental factors. The complexity of these interactions between the nervous and biomechanical systems is why, even if 2 stroke survivors have similar stroke lesions, they can display very different gait deficits. So here I review how gait is typically analyzed. On the x-axis, I have gait analysis complexity, and on the y-axis, data complexity. Physical therapists typically use visual observation and provide subjective scores or ratings on standardized gait assessment reports based on their background and clinical experience. While this approach is relatively quick and requires no special equipment, there may be reliability issues, especially between clinicians. Time-based metrics such as gait speed adds a quantitative measure of gait function. However, it is insufficient in detecting specific abnormalities in individuals' gait patterns. With the advent of technology, 3-dimensional camera-based systems can be used to quantify continuous joint kinematics, kinetics, and other variables over time. But it is then time normalized to the gait cycle, and furthermore, discrete variables at specific time points are created, such as the peak knee angle. And while these variables provide insight into specific joint mechanics, any 2 given individuals could have similar discrete metric magnitudes, but their gait patterns may be very different. The aspect of gait analysis that is much less studied is continuous gait dynamics, and this is where my research lies. Gait dynamics are the rules that govern how individuals' gait evolves over time, and not solely at a specified time point. So physics-based models and musculoskeletal models, they have been developed to simulate gait, but there remains a gap in the field, and it's appropriately accounting for the constraints of the neural and biomechanical systems. So what is gait dynamics? Gait dynamics, when I refer to it in this presentation, I'm referring to how the system changes over time. The dynamics captures information that's responsible for predicting future variables based on information at previous time points. So here's a swinging pendulum example. And this dynamical system is composed of a state, or the angular position and its derivatives, time, of course, and rules describing how the system evolves. So in this case, we have the known law, physical law of gravity, acting downwards on the mass, and first principles knowledge of Newtonian mechanics, describing how the mass would accelerate when subject to a force. So using classical approaches, such as differential equations, explicit equations of motion can be derived for this simple system. Musculoskeletal models have been developed to simulate the dynamic system of walking without having to rely on experimental data. And these biophysical models combine detailed representations of the musculoskeletal anatomy and biomechanical principles that can simulate the dynamic behavior of walking. However, there are some challenges associated with these models. There's no way to currently feasibly capture the neural control mechanisms of gait. So these simulations are based on assuming that the central nervous system optimizes some performance measure, for example, effort or metabolic cost of walking. And thus, they're framed as optimal control problems. Additionally, these models are typically subject-specific, so they do not consider the precise muscle coordination or changes in recruitment patterns with different conditions and diseases. And lastly, there are just so many parameters, joint angles, moment arms, muscles, tendons, that even with such a detailed model, simplifications are still made to estimate the full representation of the neuromusculoskeletal system. Researchers have used data-driven machine learning, particularly neural networks, to approximate the dynamics of complex systems, especially when many physical parameters are unknown. Neural networks have been used to successfully model complex biological systems. For example, in systems biology, neural networks predict protein folding, as shown on the left. And on the right, in neuroscience, they infer neural population dynamics in non-human primates. So these biological systems can be described mathematically by constructing computational models that track how the system measurements evolve over time. However, in human gait, understanding the biomechanical and neural processes governing its time evolution is challenging. However, neural networks implicitly encode latent states that influence observable outcomes, such as our body postures and our joint angles. So this implies that by analyzing measurable data like kinematics, neural networks can infer the underlying processes driving the progression of gait without needing explicit knowledge of all the influencing factors. So in AIM-1, I developed a quantitative framework that is more objective and holistic than visual observation. So it focuses on capturing multi-joint, individual-specific differences in coordination over the entire gait cycle, rather than discrete metrics at specified time points. This framework seeks to capture individual specificity, while enabling comparisons of gait dynamics across different individuals and conditions, for example, gait speed. And this is all done in a unified model, eliminating the need for separate individual-specific models as used in traditional musculoskeletal modeling. Additionally, the goal is to gain reasons behind these differences, which can potentially inform personalized gait rehabilitation strategies. So in AIM-1, I use a simple recurrent neural network, or RNN, to develop this dynamical characterization of gait. I hypothesized that the latent state variables of the RNN infers the individual-specific dynamics of human gait coordination without specifying any physiological constraints. This aim is based on published work at PLOS-Comp Biology. To test whether my RNN model can effectively capture individual differences in gait dynamics, I leveraged a data set that was collected by Dr. Tricia Kaser, who is one of my committee members. So this data set included able-bodied and stroke survivors who underwent treadmill training at 6 different speeds, ranging from self-selected to comfortably fast. Kinematic data, specifically joint angles, provide measurable insights into the progression of gait. For my initial proof of concept, I focused on gait kinematics observed in the sagittal plane, so side view. And using these measurements, the RNN estimates hidden factors or latent variables that govern the evolution of gait. All of the data across all individuals and the 6 different speed trials served as the input to the RNN model, which was trained to be able to predict a one-step time-shifted version of the kinematic input sequence. The internal model activations, denoted here as the H's and C's, they were extracted in the form of these time traces for each individual. And these inherently encode the time-varying changes in kinematics or postures underlying gait or gait impairment. So since the RNN was trained on data from multiple individuals, all individuals' dynamics are defined by the same set of model activation weights in the RNN. Using these inferred dynamics, or internal model activations of all individuals, I applied principal component analysis to reduce the dimensionality of the many parameters over time to 6 components. I then applied phase averaging to these projections to gain a representation over a single gait cycle, and I refer to this low-dimensional dynamical representation as the gait signature. Here are the first 3 principal component projections plotted in 3D, allowing us to observe that the dynamics are different between an able-bodied individual in blue, high-functioning stroke survivor in red, and a low-functioning stroke survivor in orange. Here are the 3D gait signatures of the 5 able-bodied individuals walking at their 6 different speeds. Each loop represents a speed trial with different shades of blue representing each individual. The dynamics revealed stereotyped, well-behaved looped structures with each individual's speed signatures tightly grouped together. Below, we could observe these same signatures colored by the 4 gait phases. So as an individual transitions from left swing to left stance, and then right swing and right stance. Now on the right, I show the gait signatures of the 7 stroke survivors walking at their 6 different speeds. And there's a lot more variability between individuals, but each individual's speed trials are still clustered together. We also see that there are individual specific deviations in these gait signatures across different gait phases. To better visualize the differences between all of these gait signatures, I used multidimensional scaling to project the gait signatures to 3D Cartesian points in space. And the benefits of this is that it simplifies the data, making it easier to identify patterns and relationships. It also reveals structure, so I could detect clusters now, outliers, and even trends. And the relative proximities between points in this reduced space corresponds to similarity if points are close by, or dissimilarity if the points are located further away from each other. In this 3D MDS map of all gait signatures, able-bodied individuals, or the blue shades, cluster to the left, showing dynamical similarity. Although it appears as though there are 2 clusters, this is likely due to the small sample size of only 5 able-bodied individuals. And later, I demonstrate that more samples fill out this space. Stroke survivors on the right, in red shades, they show high individual variability and occupy distinct regions of the map. I calculated the centroid of all able-bodied individuals, denoted by this gray square, as the able-bodied reference. And I used the Euclidean distance metric to quantify the similarities of gait signatures to this centroid. By using this metric, I show on the right that able-bodied individuals in blue are closest to the centroid, and we expect this. High-functioning stroke survivors in red are further away, followed by low-functioning stroke survivors, which are most distant indicating that they're least similar to able-bodied dynamics. So while this provides a way to compare overall dynamics and between individuals and groups, we still need to identify the biomechanical components of the six-dimensional gait signature. So we can take a specified number of loadings on the PCs and drive them through the trained RNN model to reconstruct kinematics. And on the right, we observe the reconstruction of kinematics using all 6 PCs, which accounts for most of the joint's motion in the sagittal plane. Using the same framework, I drive each principal component loading independently through the model to reconstruct the kinematics associated with that specific PC. So on the top right, we observe the reconstructed kinematics pertaining to PC1, and it appears to correspond to interlimb hip flexion and extension coordination. And on the bottom right, PC2 appears to infer the dynamics responsible for interlimb knee flexion and extension coordination. So we see that these dynamical components map back onto specific aspects of interlimb and interjoint coordination patterns. Here's an example with 2 low-functioning stroke survivors walking at similar slow speeds of around 0.3 meters per second. And they have similar Fugel-Meyer scores, and this is a stroke-specific impairment index score. So despite these clinical similarities, their 3D gait signatures are very different. For the individual represented in light purple, ST4, the 6 PC projections show major deviation from able-bodied dynamics in PC3, which is entirely above the able-bodied confidence interval. And for the other individual represented in dark purple, ST2, their PC3 is actually within the AB confidence interval. But for instance, PC4 in the paretic swing phase is completely flattened. So this demonstrates that despite similar clinical scores and walking speeds, the gait dynamics differ significantly. To identify specific coordination deficits in stroke survivors, we could manipulate the PCs and reconstruct the associated kinematics. So here I show an able-bodied individual's dynamics on the left panel, and ST2's dynamics from the previous slide, the dark purple individual, in the right panel. By replacing the healthy PC4 with the impaired PC4, we can observe the reconstructed kinematics of this manipulation in red compared to the original AB kinematics in black. And I show that the gait deficit reveals minimal paretic knee flexion during paretic swing, and potential overcompensation in the non-paretic knee during the same time point. While this able-bodied individual's dynamics does not reflect ST2's pre-stroke gait, this is just an example illustrating how manipulating dynamical components can reveal insights into gait coordination deficits. So in conclusion, for AIM-1, I've shown that an RNN can capture individual-specific neuromechanical gait dynamics for both able-bodied and impaired individuals within a single model, enabling direct comparisons across cohorts and speeds. And this eliminates the need for generating individual-specific models as required in musculoskeletal modeling. Gait signatures are developed without specifying any physiological constraints, enabling comparisons across diverse populations with unknown neuromechanical differences. The subcomponents of the gait signature are biomechanically interpretable, allowing us to map specific subcomponents back to multi-joint kinematics over time. And gait signatures offer a method to analyze deviations in gait coordination from normative patterns, as demonstrated with the gait sculpting framework. So the gait signatures framework shows promise in eliciting individual-specific gait signatures across cohorts, but AIM-1 had several limitations. Firstly, it was a small data set. The model was only tested on 5 able-bodied individuals and 7 stroke survivors, and a larger cohort of able-bodied gait could potentially reduce the individual-specific nature of these signatures. Additionally, we only looked at a short trial length of 15 seconds. So I wanted to determine whether longer trial lengths would account for more intercycle variability and test the robustness of gait signatures. Thirdly, there was a limited speed range. The speed ranged from self-selected to comfortably fast. And it's unknown whether a wider range of speeds would affect individual specific trends. To determine how able-bodied individuals alter their gait signatures with speed, I test the predictability of the relationship across individuals. The influence of external factors on gait dynamics across different speeds has not been explored. So previous work by Shivatal and Ting suggests that individuals use the same common neural mechanisms or muscle synergies across different contexts, such as balance and walking. However, it is unclear if individual differences in balance ability in an unimpaired cohort correlate with changes in gait dynamics between speeds. So thus, I investigated this relationship in my study. And in AIM-1, I only used sagittal plane kinematic data. So here I explored whether 3D kinematics or 3D kinetics might improve individual-specific gait signatures. So in AIM-2, I investigate whether individual specificity is maintained in a larger cohort of able-bodied young adults and how dynamics may change over a wide range of feasible walking speeds. I hypothesized that gait signatures remain individual-specific and changes in gait dynamics with gait speed would be similar among able-bodied adults and would be associated with balance ability. This work is summarized in a paper that's currently under review at Scientific Reports. Our cohort consisted of 17 able-bodied young adults walking on a treadmill for 60 seconds. There were 9 speed conditions ranging from a fixed extreme slow speed of 0.3 meters per second to individuals' walk-to-run transition speed, which is extremely fast. I collected continuous kinematics and kinetic data and tested individuals' balance ability using the narrowing balance beam test. To create gait signatures, I utilized the same RNN framework described previously in AIM-1. Gait signatures were developed for 4 different data types. So 2D kinematics in the sagittal plane, which was done in AIM-1, 3D kinematics, which now includes the frontal and transverse planes, 3D kinetics, which includes ground reaction forces, joint moments, and powers, and a combination of the 3D kinematics and kinetics. To compare whether other gait signature types would improve the individual specificity of gait signatures, I trained support vector machine classifiers to compare the individual discriminatory power of each gait signature type in this larger able-bodied young adult cohort. I found that individual differences in both 2D gait signatures on the left and 3D kinematic gait signatures on the right were consistent across the wide range of walking speeds. For instance, all 9 of YA17's looped speed trials in olive green are clustered together. And then higher order PCs 4 to 6, they show even more individual-specific clustering, especially in the 3D kinematic gait signatures to the bottom right. In a study using SVM classifiers, I showed robust classification of kinematic gait signatures across speeds by varying the number of speed trials per individual in the training set. So on the right, with just 1 trial per individual in the training set, over 90% accuracy was achieved using 3D kinematic gait signatures, demonstrating consistent individuality across speed variations. Perfect individual classification accuracy was achieved with 3D kinematics when using 4 or more random speed trials per individual in the training set. To the left, 2D kinematics achieved 96% accuracy with only 4 random speed trials per individual, indicating that there's a substantial individual-specific information that can be attained from sagittal plane motions alone. The higher accuracy with 3D kinematic gait signatures suggests that there are meaningful differences in frontal and transverse plane kinematics among able-bodied young adults. And this supports that one should consider gait variables beyond the sagittal plane, especially when analyzing patient populations with known deviations in these planes, like stroke survivors. Note here that 3D kinetics had lower individual classification accuracy than 3D kinematics, and it was around 97.8%, I haven't shown it here, suggesting that adding kinetic information does not enhance discriminatory power in gait signatures. This aligns with previous studies that use kinematic data to infer kinetic data, emphasizing the established mechanical relationship between the two. To test whether individuals exhibit similar linear changes in dynamics across speeds, I used linear mixed effects models to predict each trial's 3D MDS coordinates from trial speeds. Despite individual specific differences in gait signatures, we observe consistent directional linear changes in 3D MDS representations of gait signatures with speed across participants. And hierarchical bootstrapping results, which are those many lines per participant, has confirmed the robustness of the LME models to variations in input data. To test whether gait signature changes were related to an external measure of individuals' functional ability, I tested individuals' balance performance on a narrowing balance beam test. And I found that individuals with better balance or higher scores alter their gait signatures more, from self-selected to extreme slow. Secondly, participants with faster self-selected speeds exhibited better performance compared to those with slower self-selected speeds. And the habitual walking speed preferences observed among able-bodied young adults may be linked to their individual balance capabilities. Further, individuals with slower self-selected speeds change their gait signatures less between self-selected walking and extreme slow speeds. So that means they were less able to modulate their gait dynamics. It's important to note here that balance ability may be impacted by several factors, such as specialized movement practice. So for example, ballet dancers or gymnasts, they are likely to have better balance. And also, there was a lot of competitiveness from participants on the balance beam, as they actively tried to beat their previous balance beam scores, and this may have also impacted our results. In conclusion, for AIM-2, kinematic data effectively captures individual differences in gait dynamics among able-bodied adults, suggesting the potential applicability in heterogeneous populations like stroke survivors. Able-bodied gait signatures maintain consistent dynamics across speeds, enabling reliable identification of individuals regardless of walking speed. And this may be valuable for biometric recognition and research applications. Gait signatures show a linear relationship across various walking speeds, offering insights for sports training, perhaps, for identifying specific speeds that would elicit target dynamics. And lastly, individuals with better balance demonstrate more flexible modulation of their gait dynamics across different speeds, suggesting that the ability to more independently control their muscles influences speed-dependent gait adjustments. So now that I've shown you proof of concept of the gait signatures framework, the question remains whether this framework holds any clinical utility. Current clinical evaluation methods lack the ability to effectively quantify treatment responses to interventions. Clinicians often rely on gait speed and discrete biomechanical metrics or targets, such as paretic push-off, to assess treatment response. However, what happens when improvements in one metric comes at the expense of another metric, impacting overall gait quality? This third aim seeks to explore whether gait signatures can provide a more integrative approach to whether interventions yield beneficial or detrimental effects on overall walking dynamics in stroke survivors. So aim 3 investigates this clinical utility of gait signatures to track treatment efficacy in 2 gait interventions, fast and fast functional electrical stimulation in stroke survivors. And I'll explain these 2 interventions in another slide. I hypothesize that gait signatures of stroke survivors who respond positively to intervention, and by that I mean improvements in their gait speed or clinical biomechanical targets, they would not necessarily move closer to the reference able-bodied centroid. This work is currently under manuscript preparation. So fast training involves progressively increasing the treadmill speed across sessions. This aims for high-intensity bilateral stepping practice, and clinicians measure improvement of this intervention by their walking speed. So they use the 10-meter walk test. Fast FES is a gait rehabilitation paradigm that encompasses fast walking plus muscle stimulation. Surface stimulation pads activate paretic dorsiflexors during paretic swing and paretic plantarflexors during late paretic stance to promote improved gait function in stroke survivors. Previous studies show that fast FES improves key biomechanical outcomes like paretic push-off, trailing limb angle, and ankle angle at initial contact. So while fast training often boosts gait speed, this alone does not necessarily indicate recovery. It may instead promote compensation or reinforcement of poor gait quality. And in contrast, fast FES targets neurological recovery pathways to enhance paretic propulsion, but its impact on overall gait dynamics remains uncertain. I created gait signatures for 6 stroke survivors undergoing fast training and 6 undergoing fast FES training, and each comprised of 12 sessions, so 3 sessions of training per week for 4 weeks. Continuous 3D kinematic data was collected at specified sessions. So we collected at baseline, which was before the intervention started, sessions 1, 4, 7, and 12, and then post 12 sessions of intervention, which I will call P12. And all the individuals were tested at a fixed self-selected speed that was established at baseline throughout these sessions. I also used the same data set of 17 able-bodied young adults from AIM-2 as an unimpaired reference. So recall that able-bodied young adults from AIM-2 walked at extremely slow speeds, but now it's matching the speeds of some stroke survivors in the data set. So despite similar speeds, able-bodied gait signatures, or those gray circles, are distinct from stroke survivors in the colored circles. And this is supported by a high support vector machine gait group classification accuracy of around 99%. At very slow speeds, some slow able-bodied gait signatures, so those small gray circles, intermingle with stroke gait signatures, suggesting that when able-bodied individuals walk extremely slow, their dynamics more closely resembles that of stroke dynamics. On the right, self-selected speed gait signatures represent typical able-bodied dynamics. So the able-bodied centroid, or that gray square, was calculated accordingly. Individuals showed varied progress over training sessions. For example, if I could direct your attention to this dark purple individual, or ST30, we see that there lacks a clear progression in their rehabilitation journey. So factors such as general health, comorbidities like cardiovascular diseases, motivation, and even measurement consistency despite efforts to standardize during data collection, these may all contribute to variability across these sessions. So this framework shows promise for tracking rehabilitation progress in gait dynamics. However, further research is needed to understand how specific regions of this gait map are associated with potential barriers that may alter stroke survivors' intervention response pathways. In stroke data, variability throughout training can be complex. So let's simplify and focus on the changes from baseline to P12 or post-12 sessions of training. In the left plot, baseline gait signatures of all 6 stroke survivors in the fast training group shifted closer to the able-bodied centroid, so there was smaller magnitude Euclidean distance post-training, suggesting that greater similarity between the stroke and able-bodied gait dynamics occurred with fast training. Now in the right plot, in contrast, only 2 out of the 6 stroke survivors in the fast FES training group moved closer to the able-bodied centroid post-training. So let's identify responders to gait intervention based on changes in gait speed. So this was measured based on the 10-meter walk test before and after intervention for fast individuals on the left-hand side of the plot and fast FES on the right-hand side. The dashed red line here represents the minimal clinically important difference, or MCID, for gait speed, which is about 0.1 meters per second. In the fast group on the left, 4 individuals responded positively, denoted by these yellow stars, while in the fast FES group on the right, 3 individuals responded positively. Responders generally moved closer to the able-bodied centroid with training compared to non-responders in the plot on the right, although this observation is based on a very small sample size. Some outliers were evident. For example, there was 1 responder identified by gait speed, which moved away from the able-bodied centroid, and 2 non-responders moved towards the able-bodied centroid. So this highlights a complex relationship between treatment response and changes in gait signatures. Discrete variables provide specific insights into gait changes. And here are the pre to post intervention changes in 3 biomechanical targets of fast FES. The minimal detectable change, or the MDC, quantifies the smallest meaningful change above measurement error, denoted by these red lines. 4 individuals in the fast group and 1 individual in the fast FES group on the top plot responded positively to paretic anterior ground reaction force, or push-off. In the middle plot, no stroke survivors in either group met the MDC criteria for trailing limb angle. And in the bottom plot, no individuals in the fast group responded positively via the paretic ankle angle at heel strike. In contrast, 4 out of 6 of the stroke survivors in the fast FES group showed improvement. However, 3 out of these 4 of the responders moved away from the able-bodied centroid, despite improvement in this variable. And this suggests that while improvements may occur in specific biomechanical targets, overall gait dynamics may not approach that of able-bodied gait. Ultimately, individuals respond uniquely to treatments, highlighting the need for personalized approaches in gait rehabilitation to enhance effectiveness and outcomes. In conclusion, for AIM-3, gait signatures track changes in stroke gait dynamics during fast and fast FES training, offering an integrative method to capture treatment response in multi-joint coordination over time. Improvements in discrete variables may not align with gait dynamics becoming more similar to able-bodied dynamics, indicating potential trade-offs between different clinical metrics that impact overall walking dynamics. Reflecting on my thesis, I've made several contributions to science, engineering, and potential clinical practice. In AIM-1, I have demonstrated that individuals' gait dynamics are unique, and they remain individual-specific across different walking speeds. This provides a generalizable framework to add to the engineering toolbox to be able to identify and interpret differences in individuals' dynamics regardless of the speed that they walk, and without having to specify any physiological constraints about the individuals. I contributed clinically in AIM-1 by demonstrating that the Gait sculpting framework, or the manipulation of those PCs, may help identify specific coordination deficits in stroke survivors. In AIM-2, I have demonstrated that able-bodied young adult cohort speed-induced changes in gait dynamics are consistent across individuals, and the changes are approximately linear. And this may have implications in sports training. I show that better balance may be associated with higher flexibility to modulate gait dynamics across speeds, which uncovers potential factors influencing gait modulation, which can have potential implications for the development of tailored rehabilitation strategies. And in AIM-3, in the clinical sense, I demonstrated a proof of concept supporting the use of gait signatures as a tool to quantify gait quality more holistically than discrete metrics, and I show that gait signatures can be used to track treatment-induced responses to different interventions. This work lays the foundation for further research in understanding human gait dynamics, and future avenues include exploring alternative model architectures for gait signatures, such as using variational autoencoders, for example, which may improve individual-specific representations, as they're known for having better control over the latent space and reducing tangling of latent dimensions. Also investigating alternate dimensional reduction techniques beyond PCA. So for example, functional data analysis may uncover dynamical subcomponents that are more directly aligned with clinical functional aspects of gait. Additionally, somehow integrating multimodal gait signatures, for example, EMG, recording from the muscles, or EEG, recording from the brain, and additional clinical insights can enhance our interpretability of differences in individuals' gait dynamics. And since experimental data sets are typically very limited by small sample sizes, enhancing training data with synthetic samples may be beneficial. So potentially using physics-based simulations or generative adversarial networks can simulate a range of data spanning features like diverse body sizes or different heights or conditions that may not be fully represented in small experimental data sets. Additionally, given that machine learning models are generative, they could potentially predict the time evolution of individuals' responses to particular gait interventions, which can reduce trial and error intervention testing in clinical settings. And finally, integrating gait signatures with musculoskeletal models could potentially isolate mechanisms influencing gait dynamics. So synthetic data spanning parameters like joint stiffness or hip flexors or muscle co-contraction can be used to generate synthetic gait signatures that could enhance our interpretation of the gait map landscape. With that, I would just like to acknowledge all the individuals truly that have contributed to this work, my 5 committee members, and I'd like to thank all the funding sources that support this work. I give a lot of thanks to my advisor, Dr. Lena Ting, who is my biggest advocate in grad school, and she has allowed me the opportunity to explore very multidisciplinary and exciting research in a lab. And she always connected me with key individuals that would help drive the research forward and also inspire my future career paths. Above all, she understands that while research is necessary, balance is important. So she ensured that my time in her lab was not only productive, but filled with wonderful memories that I'll cherish forever. A special shout out to my 2 unofficial co-mentors, Dr. Kesa and Dr. Berman in the front here. They opened their labs to me and provided me with resources and comic relief and support and invaluable guidance throughout my entire PhD. A big thanks to Kanish Jain, he's in the Berman lab, whose own research inspired this work, using RNNs to gain latent space representations of dynamics. And he provided countless hours of technical assistance and support during the early years of my PhD. And to my postdoc mentor, Dr. Michael Rosenberg, in blue, for all his support, guidance, and both detailed and useful feedback during the later years of my PhD. And lastly, I can't forget my beautiful family, friends, and lab mates who have all helped me in positive ways throughout my PhD journey. I thank you all for being here, both in person and online. I thank you for your attention, and I welcome any questions that you may have. Thank you.
Spend a few minutes to take questions.
Yes.
Amazing talk. That was really outstanding. I acquired a lot of knowledge from that talk. And it struck me at the very end when you were talking about your future directions. I was really curious about two things. One, if you looked at how reliable these signatures that you develop for an individual are across sessions, because of that variability in treatment response that you mentioned, is it actually that they're responding to the treatment differently over time, or is it just that in these individuals who are maybe slightly variable and heterogeneous in the first place, that it's just not able to track that well, even if there was no intervention? That's the first question. And then the second question is, I was interested in the linear relationship between the speeds or how they were able to modulate speeds and their balanceability. Do you think that would be a good predictor in individuals with neurologic disorders like stroke if you had, say, a measure of balanceability that you could predict who would maybe more likely respond because they seem to, it would be like a clinical proxy for their flexibility and being able to perhaps modulate or change their dynamics? Just a couple thoughts on that, or if you even looked at that.
Sure. Yeah, thank you. Thank you for your question. So the first one was asking whether I tested the reliability of Gait signatures across sessions so that we can confirm whether the changes we observe in stroke survivors with intervention were meaningful changes and not just perhaps data collection issues across sessions. And unfortunately, I did not test that. That is something that I think, looking back now, should be tested to make sure that these changes that we see are reliable. However, we do see across the board changes in these stroke survivors that are very different in different ways, and I could think that they perhaps are related to the fast FES intervention, but I do think that method that you recommended is worthwhile. With respect to the balance question, which asked whether balance beam score could potentially be a predictor of whether individuals would more flexibly modulate their gait dynamics. I think more testing needs to be done. However, this does show great promise. And since muscle synergies work, which looks at individuals with less muscle synergies have worsened balance, and as we see here, for able-bodied individuals, at least, individuals with less ability to balance or poor balance scores, they don't modulate their gait dynamics very well. So we have to test it in stroke survivors, but I think it is promising here that you can use that framework, thinking that with higher number of muscle synergies, you have greater independent control of your muscles and therefore perhaps able to modulate better. But yeah, I can't make any claims on a clinical cohort based on an able-bodied study right now.
Thanks.
Yep.
Awesome talk, Taniel. And you found that 3D kinematics was better than 2D kinematics. Do you have a sense how much added effort is it? My guess would be that it comes from the hip. Do you really need 3D kinematics and neuromechanical to improve your model?
I would say it depends. I think certain cohorts, like for example in stroke survivors, they may have issues with inversion or eversion in their foot. So I think 3D kinematics would be able to detect that change. It may not be across everyone, but I think it's important to include it for those edge cases who may have issues with their knee, perhaps in other planes like the frontal or transverse planes.
The other thing that was striking or surprising was that kinetics didn't give you much more.
Yeah, so it didn't give much more in terms of the individual specificity. But I do think kinetics could be important for different applications where kinetics is important. For like exoskeletons, for example, like tracking joint movement over time could be insightful. So if someone was interested in that application, using kinetic signatures might be a more feasible approach.
Did you explore, consider whether those 3D kinetics provided more information when comparing the individual stroke compared to the body?
Unfortunately, I did not.
So I have a question about aim 3.
Mhm.
I think this is like selfishly for me. So for aim 3, you're interested in like fast versus fast FES. What's the timing of when you actually captured the walks after fast FES? And like at fast, the FES was on when you were capturing for both.
So just to clarify, we did not collect data during training. Right, so it would be right after their training, about 30 minutes of training. I think they might have had a 5-minute break. Then their data collection would have happened, just looking at how they walk at their self-selected speed that was determined at baseline. In terms of post-12, how far out that was from their last training session, that's variable. And I think that depends on individuals and their schedules and stuff like that, but yeah. Within a week, according to the... No. Yeah, I explained that. Yeah. Yeah, of course. Okay.
Questions? First from Lunchain. 2 questions. First, you showed different gait signatures among your stroke patients. I was wondering if you use any clinical evaluation tools to see if these patients' gait can also be differentiated without the use of complicated machine learning procedures.
Okay, so could you just repeat the question?
I think he's essentially asking, do you need gait signatures or could you differentiate between these people using clinical assessments?
Okay, so I guess my answer to that is I believe that gait signatures should be used, not necessarily in its current framework, but some aspect of it to give a more holistic or integrative approach to considering multiple aspects of individuals' movement over time. I think clinical metrics could be very coarse. So for example, looking at gait speed, it doesn't tell me any other information happening in different parts of their joints over time. And then as I showed, let me go back to this slide here. So for example, these are clinical metrics that are used to assess interventions. And as you can see, there are variable responses. Someone may improve their paretic push-off, but then their angle at heel strike is, has poor quality. So gait signatures just offers a way to kind of encapsulate all of the information so that we can make judgments based off of a more integrative assessment. Second one, yes.
So if the gait signature of a stroke patient is not getting closer to a healthy control after rehab, how should a clinician do, or I guess what should a clinician do about this in terms of their treatment plans and what areas they should work on?
Yeah, so that question could be answered only when we do future work to really map out this gait signature space. So for example, if an individual or stroke survivor is not moving towards the able-bodied centroid and their pathway is being directed elsewhere, really understanding the physiological mechanisms in each part of the map, like shown in the future directions, that would help a clinician, for example, know where to go next. That is the hope. That is definitely in the future and not conducted at this time in my research. Oh, okay.
Okay, this might be incredibly naive, but if I remember correctly, it looked like across the able-bodied subjects, the variability was really in PC3.
the most. And so I'm just curious about your use of the Euclidean distance as a measure to progress towards healthy. Whether it's the Euclidean distance that we should be looking at, or maybe just looking in one plane in that space.
Yeah, that's an excellent question. So the question, I'll repeat it, is asking whether the Euclidean distance was necessary or whether other metrics could have provided different information, perhaps.
Target to
Yeah.
minimize.
Yeah, so my use of the Euclidean distance was just a simplified way to get at the most direct pathway to able body. I do agree that looking at maybe individual axes, for example, using Manhattan distance metric, which accounts for how much distance was moved in X versus Y versus Z, will probably give a different answer, but I didn't test a different metric. Yeah, so it will. Thank you.
One more question
One more from chat, and this is among many, many congratulations.
Oh, thank you.
This is from Sam Hawk. I was wondering how generalizable the framework may be for other clinical assessment tasks, such as timed up and go or the 4-score step test.
Okay, so the question is how generalizable the framework is in terms of well, you're not the person asking the question, but I'm thinking in terms of comparing to gait speed like I did. So how would slotting in time up and go work?
I think Sam's asking, could you apply gait signatures to these
Oh,
tasks that are less rhythmic and have these transient periods or something like that? More complex.
currently, no. Short answer, but the gait signatures framework really hinges on using periodic data because we do this phase averaging step at the end. But what it's really getting at is learning the underlying dynamics, and that occurs with having a set task with periodic data. So if there's a trial with changes in acceleration or speed or different things, I think that might impact results and may not be generalizable in that case, but that's yet to be tested.
Okay, we should stop and let's congratulate the. Thank you all for being here. There's still some donuts and coffee, but I'm going to take you out of the room.
How are you? Thank you.
Michael ate that shit up.